properly resolved by our fantastic technical team. So, hi everyone, and welcome to the first seminar of the 2025-2026 CAB seminar series, where we'll focus on championing the research of the CAB. So this series will focus on the CAB researchers. And to kick things off, we've got our director, Víctor Parro Garcia, who is an expert in the field of astrobiology. So Víctor started his career at the Universidad Autónoma de Madrid, where he studied molecular and environmental microbiology. And since then, Víctor's become a sort of leader in the field of astrobiology here at the CAB, where he's been here for over 20 years. And sort of in that time, he's become the sort of associate editor of the Astrobiology Journal, and he's a member of the N-fold NASA coordination group. So Víctor's research focuses on the presence and preservation of microorganisms in astrobiological environments. And to that end, he's led sort of multiple international projects, one of which is SOLID, the Signs of Life detector, which has had great successes in the field, where Víctor's led expeditions to places like Antarctica, Greenland, and the Atacama Desert to exploit the capabilities of SOLID. So with that, I'll sort of pass over to Víctor and say, thank you very much, Víctor, for being the first in this series, and I'm really looking forward to the seminar.
Okay. Thank you. Many thanks, Lee, for your presentation. You're very kind. And well, the idea of this new series of seminars, as Lee said, is that we present our work here in CAB, because I think we have a lot of things we have done over the years, and we are still doing now with our projects. And I'm pretty sure, because it's even my case, there are many of us don't know exactly what our colleagues are doing here. So I think it's very important just to start this series of seminars, and not necessarily the PIs of the project, but whoever in the project that can explain what the project is dealing with, or what are the objectives, or how is the state of the project. So the idea just, well, I offer myself to start and to set an example. I don't know if it's good or bad, but at least an example to start this series of seminars. And please, I encourage all of you that has any responsibilities or not in the projects, just to explain to all of us, what are you doing, and what can we, how can we collaborate or interact each other? Because, by the way, one of the questions of the other days, external advisory board that we have here at CAB was, how do we deal with transdisciplinarity? What we do to promote and the transdisciplinarity and interaction with all the groups. And this could be one of these activities, must be one of these activities. So, well, I'm gonna give you the state of the project SOLID that you have heard perhaps along the corridor, or sometimes you have heard in many talks about the project. SOLID stands for Signs of Light Detector, and we initiated the project by 2001 or something like that. And the basic idea is just to search for molecular biomarkers in the environment, in environmental samples, using, in our case, an antibody macroarray. An antibody that, for all of those of you that are not familiar, antibody macroarray is a small surface of support, a microscopic slide or whatever, where you can immobilize 100, even 1,000 of reagents. Could be DNA, could be antibody, could be proteins, could be even cells, whatever, and make them react and give a signal. Okay, so the idea was to put in one of these sensors a lot of antibodies that can capture whatever signal we are interested to find. Okay, later we will explain what are the strengths of this and what are the weaknesses that already have, for sure. So first of all, I wanted to many of you to think about what is a biomarker, what we understand a biomarker. At least there is always a controversy into or using biomarker or biosignature or well, whatever the case. As I understand this, a biomarker could be considered as a substance that can link a biological process with this substance. Okay, so there must be a connection with the biological process. Otherwise, it's just a molecule that is wonderful, but we don't know if it is a real true biomarker or not. Or in a more general way of speaking, we can say that we consider, at least for us, a biosignature, other type of things, like, for example, mineral biosignature, those minerals that can be produced through the action of biological metabolism, etc., etc. Okay, or for example, as you can see the bottom picture, you can see the stromatolite structure that is an indirect result of biological activity. Yeah, but how can we detect? Okay, is when we see some, you can see some animals, you can see whatever. Okay, this is life. Yeah, for sure, it's life because we know this life because we have been very familiar with it, we've been living with it, etc. But it's not that easy when you see other structure. For example, you see at the bottom left, you see, what is this? One could say, okay, this is a building, a mountain, or whatever. No, but this is a biosignature. This is a termite structure. So it's being done by living beings. But if you don't know this, you have any idea. We have to, if we have not these macro signals, we have to go deep inside and searching for structure of biomolecules that are better preserved in the geological record. For example, through biochemical structure, proteins, DNA, whatever that can be preserved a long time, or the most stable type of molecules that are lipids in this case. In this case, you see at the bottom right, is opanoids or the triterpene structure that are very stable, and these structures form part of the membranes of the bacteria. These compounds are similar to the cholesterol in eukaryotic cells, and they're abundant and they can be preserved. And it's a molecule that so far there is no way to produce from a biotic from a biotic process. Okay. So yeah, it's not that easy, and this complicates the search for life in astrobiology. So far, the technology has been used in the different astrobiological missions, like the Vikings, or even the mission that are now on Mars. They use basically the GCMS, the gas chromatography mass spectrometry techniques for detecting volatiles. Why that? Because preparing the sample for analysis millions of kilometers apart is very difficult. So you have to do the simplest way you can do it. One way just to hit the sample and capture the volatiles and analyze by GCMS. And this is the most, but you have many problems. It's very easy to produce, but you have very inefficiency in the extraction, just volatiles. Many compounds still remain bound to the mineral substrate. And also many of these compounds are destroyed, so you can burn it with heat. So at the end, you lose your biological, if any, activity information. Okay, so it's very complicated. There have been proposals other techniques like capillary electrophoresis, etc., but it still is not easy to implement for space technology. And some of the instruments that are now on Mars, like the ones in Perseverance, is the Raman spectrometer, is the Sherlock, that detect the presence of organic matter, just organic matter. They can't tell you anything else if it is what kind of molecule, if any kind of biosignature or whatever. So there are other, as I said, well, there are new instrumentation that are on the way, like the MoMA instrument that is let's hope that can be launched in 28 in the Rosalind-Franklin mission. It's similar to the in the end, it's a a GCMS also, but they also have what is called the laser desorption. This is hit a laser, desorb the molecules. But again, these techniques has a lot of, I would say, inefficiencies in the sense that they are not able to extract everything they can. Okay, but yeah, and in the end they can break the molecules and lose information. What we proposed some time ago is, okay, well, for sure, all techniques are good. All of them have their own field and they provide information. What we suggest is, okay, for those molecules or compounds that you are not able to extract or are not volatiles, or you can break with a laser or whatever, well, we can use a bioaffinity system. That is the system that uses antibody and antigens in our immune system for recognizing each other. So antibodies is, in that case, wonderful because it can detect from amino acids to nucleotide molecules to whole cells. They are the targets, they can bind it, and they don't need to even to extract from the mineral. We will show you later some result that we can detect compounds that are bound to mineral surfaces, and we don't need to remove from the surfaces. Okay, so in that case is good. But even in that situation, we had to have a general picture of what the field we are facing is. The point is, there is I don't know if, okay, there is a place where molecules can overlap because they are very, very simple. We have a place where we have amino acids, we have small hydrocarbon molecules, etc. But these molecules can have a probiotic origin too. So if we detect amino acid, it not necessarily means that there is life. It depends on what kind of amino acid. There are all other parameters that we have to take care of, the enantiomeric selectivity, etc., etc. But this region of the different space environment is the most complicated because these compounds can be shared from different origins. So for sure, when we move to the right with increasing complexity and sizes, we have macromolecular structure that if we are able to detect them, for sure, the only way we know that we can have these molecules by a biological process. So that means that the bottom left may come from life by degradation or may come from abiotic origins, what is something that we have to deal with. So the hypothesis then we started with was, okay, understanding the traces of life in nature, we can design our weight, in that case, antibodies for detecting these compounds, okay? The advantage against the GCMS is that one of the problems that has been reported, now they are trying to solve it, and it looks pretty well, but not necessarily the best. GCMS, as I told you, need hitting the sample, and hitting the sample may destroy, because one of the things that there is on Mars is perchlorate salts. There are a lot of relatively high perchlorate concentration, and perchlorate is a highly oxidant compound that in the presence of heat burn the molecules. Okay, so that could be one of the reasons for the no detection of organic matter by the Viking GCMS or many other GCMS that is still there, but they have very broken molecules and complicated interpretation. So our objective was to develop an antibody microarray to detect the higher number as possible of targets, and the methodology we envisioned by them was developing these antibodies, what is what we call the LD chip, the light detector chip. This is a part of the project and the story, and the other one is an instrument that can do that, that can do the immunoassay automatically. Not only the immunoassay, but the extraction of the compounds that can be in the samples, okay? And then validate this in the laboratory and field campaigns, and for sure propose this methodology in different opportunities for instrumentation. How the system works? Well, we have the LD chip can be a small surface where we immobilize the antibodies, this Y form, and then what we do is crude extract from soil, rocks, whatever, and this extract is filtered because we need to filter a little bit, just filtering. The idea just the procedure is to make it the most simple as possible because it has to be operated remotely, as it's not that easy. So just extract, we do by ultrasonication, filter, incubate with the antibody microarrays, then we add a mixture of the same antibody that has been previously labeled with a fluorescent tag and incubate with the chip. And in those spots where each antibody has 150 or 60 micron sizes spots, okay? So we're talking that in a small space we can put 100, 400, 500 spots, okay? If something has been retained like this yellow stuff there, maybe a biomarker, a bacteria, whatever, the label antibody can bind to free sites and then forming a kind of sandwich. That's why we call this sandwich immunoassay. Then a laser excites the fluorochrome and a CCD camera take a picture, and the picture have like this some fluorescent signal that can be quantified and analyzed. So the idea is just to have information from the dirt to data, just we can quantify, we can do our analysis and extract the information. So that the identification of the biomarker is relatively easy because we have the antibodies that we have used to produce to detect this type of compound. So if there is a positive, that means there is a direct correlation, okay? So how we select the targets for our antibodies? So this is a scheme that we developed at the beginning. So the idea is to extract natural samples or microbial isolated, then fractionate these compounds, or even by commercial companies, and then produce the antibodies. And that's we've been using since many years. We use rabbit antibodies, and then one of the ideas is, okay, let's validate this. Let's say what we are detecting with our antibodies. So all you see on the right are electrophoresis yield, where we fractionated all complex extract, and all these strips you can see correspond to true signal that are recognizing our antibodies. So our antibodies are recognizing proteins, polysaccharides, complex. In the bottom one are in that case are lipid, glycolipid complexes. So this black spot are indicating that our antibodies are working on detecting this type of compounds, okay? This type of compounds, I talk polysaccharides, glycolipids, proteins, even DNA. Some of our antibodies we have can detect single-stranded DNA and some mutation that DNA can have. For example, thymidine dimers or this type of thing can be also detected with this. This is just to illustrate that microorganisms produce a lot of biomass in the environment. This is a cell that is bound to a mineral surface that is producing all these amorphous stuff you see are polysaccharides, exopolysaccharides that are producing to interact with the environment. The microorganisms, when they are living in the environment, I used to say is they produce their own house, their own coats. If there is cold, they produce their cold. How? Producing polysaccharides. If they need water, they produce also polysaccharides to trap humidity from the atmosphere, etc. So they produce a lot of things and are kept in the environment. For example, these are biofilms from the Rio Tinto. Most of it is polysaccharides. There is a lot of mass there and a lot of cells too. So when they die, they remain in the environment. Many of the material is degraded, but many of it still remains there. So we produce a lot of antibodies against covering the most important phylogenetic groups in bacteria, from alpha, beta, gamma, delta proteobacteria, and nitrospira, actinobacteria, cyanobacteria, a lot of them, okay? This is the phylogenetic tree. Also produce antibodies against well-preserved proteins correspond to very ancient metabolisms, like could be the sulfur reduction, the nitrogen fixation, methanogenesis, etc. Okay, so we have now more than 400 antibodies in our collection. We tested all of this. This was when the chip has 200 antibodies. We tested with natural samples. We made a map testing the different antibodies and the different samples, and we see this diagonal that corresponds. That means that each antigen has a preferred reaction with their antibodies. But there are also cross reaction with others, which is not bad. It's also good because we wanted to detect things and to have to be sure that we are detecting things. We also determine the limit of the detection of each antibody. On the left, you see the detection limit for antibodies for some proteins. We are always at the level between. It depends on the quality of the antibody. Not all are working at the same way, but we can say that for protein we are detecting from 1 to 10 ppbs, and for cells it depends the type of cells, but we're detecting in some cases for cyanobacteria we've been detected even 100 cells per milliliter, which is not bad. It depends always on the quality of the antibody, for sure. We implemented the chip in the instrument. The first prototype was the SOLID-1. The idea was to demonstrate, was a prototype to demonstrate the system. We have on the left, you see, this the camera, the laser. We have different cells, and this is the chip. And the idea was to demonstrate that we were able to do it automatically and remotely. Okay, so well, in that case, it was not remote, just automatic, which was important. Okay, this was SOLID-2. SOLID-2 here, where we have 18. Too young, or too old now. And 19, I don't remember the name of 18 modules that are self-contained. SOLID-2 was tested in the 2005 campaign during the Marte project. It was the first project that the CAP had with NASA Ames in 2003. So we tested in the robotic platform. They took sample from automatic drilling, moved through a robotic platform, and feed SOLID in that case, and we analyzed the samples with some of the result, and it was already published. This is the test of the SOLID number 3, the version number 3. We split the sample preparation unit. That's the unit that extract the sample by ultrasonication filter, and then this feed the sample analysis unit where we have the chip, the antibodies, etc. Okay, but this is split in 2 units, and it was tested in 2018 or 19 in the Atacama, again in collaboration with people of NASA Ames that were involved. Well, they developed the drilling system for taking the samples. And the chip has been tested in many campaigns, this one, for example, in the IPBSL project, the advanced grant led by Ricardo in 2012, I think, where we detected for the first time there in the field laboratory, we detected the presence of cyanobacteria in samples like this, taken from 400 to 600 meter depth. Also, we have tested in many places, like here is in the Andes, in some extinct geysers. As you see here, we extra sample with the drill, and you can see how similar are those samples. I don't know if the geochemistry is the same, but at least the aspect is very similar to the one that has been done now on Mars. But the analysis of the upper sample from the Tatio with the chip, as you can see there, give interesting result with microbes that is not strange that to be there, like Acetobacillus caldus. makes sense to be there. Okay. So the present project that make the title of the talk is what we call past and present sign of life. The idea here is just to first, well, there are three main objectives. One is to increase the technological level of the instrument. Now we can say that it's around, well, TRL 5. To be TRL 6, some of the component has to be tested in environment, in vibration and other very stressful conditions. But okay, the idea is just to keep increasing the maturity, because it's not perfect. There are many things that they still are not the best performance, like the optical setting is not the best, the sonication has to be improved, etc., etc. So, but the idea is to keep on that. And one of the thing that was still pending is, you can imagine how we receive the samples and how do we measure the sample we're going to analyze. So this is one of the things that we are doing during this project. Is we have developed a new module that is called the sample receiving and delivery module, where we receive the sample from the drill, just a scoop, leave the sample without measuring anything, whatever it is, and we have it underneath. You will see later. We have a kind of a scoop that take the sample and just dosify the sample we want. Okay, we will see later. And this is from the technological point of view. Later, from the more scientific point of view, the idea is to search for biomarkers in all samples. That's why the idea is to go back in time, and one way is to make a molecular inventory of many of these samples. They mainly focus on lipids and peptides. Lipids, we have Laura that will give a talk soon, so we will leave lipids for her, and we will concentrate on peptides and proteins. And later, the idea is to use a theoretical approach is using ancestral protein reconstruction to go back in time and try to figure out what kind of protein or peptide could have been in the origin of life on Earth, that perhaps was similar to those on Mars. Okay, for sure, this is all the hypothesis, but this is a good exercise that is working very well. And for sure, the idea is to try to do new versions or new improve the technology for detecting biomarkers. And the idea is to do others, like we have been trying with a new sensor based on impedance, but has not worked very well. But we are still working on other ideas, or even using the chip for other things, like monitor the astronaut health for the moon or the space station. So we have developed some antibodies that can help us to do that. So, okay, these are the major objectives. One of starting with the technology, this is the new model that we have implemented. I think there is a video. Yes, so the idea is just, okay, you see here, this is the funnel, the big cycle. The sample drops, and underneath there is a scoop, you see? When it approaches to the extraction cell, opens, and the sample drops. That's the idea. And because the scoop has a certain capacity, is the way we dosify the sample, okay? No more than 1.5 grams, something like that, okay? And this is one of the things we have been testing in the last campaign in the Arctic, with also in collaboration with NASA Ames, in a project led by Brian Glass. They test the drill, as you see here. The drill, as it's retired, the drill, the cuttings drops into, in that case, they're using a bag just to collect the samples and analyze deeper here in the lab. And you see here how the solid, now this module is on top of the sample extraction, sample preparation, so the sample preparation unit of solid. All packages is there. So once the sample drops, we start the process, the sonication, etc., etc. And you will see how the sample drops. It's moving, You see the scoop, and now it's approaching to the extraction cell. As you see how it drops, okay, the sample drops to this, and now is closed with the piston that hermetically close the extraction cell. Then we add the liquid and do the sonication, etc. So this is the new one that's we have done. And this is just to show you one of the results we obtained directly. This summit from one of the samples taken at 50, 60 meter depth. The upper histogram correspond to the negative control. The lower one correspond to the sample. What we have on the x axis is the number of the antibodies. Okay, 200 antibodies are there. And what we have in the y is just the fluorescent intensity, is the quantification of the signal. Okay, and those signals you see here correspond to the sulfur-vibrio, Jobatter, sulfur reduction, metaproteomics. It makes sense because it's a place with a lot of sulfate, and deeper 600 meters, that means that these microbes are sulfate reducers, so it makes sense to be there. And many signals corresponding to cyanobacteria, and as Sergio and Miguel told us that there are many cyanobacterial on the surface of the site, so it makes sense to be there. Okay. So, but still keep feeding the antibody with the chip, mostly to refine the system. Why? Because one of the most relevant critics we have everywhere in every congress, everybody said is that it's too Earth-centric. That means we're producing antibodies against things that we know are here on Earth. Okay, and I say yes, true. It's authentic. The only life we know is the only thing we are producing. So okay, but even in that case, we are trying to mitigate this criticism. So one way to do that is just to develop antibodies to target other things. For example, at the bottom here, you see we have produced, and we are now a paper under review, antibodies that can detect peptides, synthetic peptides that can be produced abiotically or whatever way, that are made of D and L amino acids. So that means, well, this is a universal molecule. I don't know if all life on Earth has peptides, but if they have peptides, it must be like this. We are also producing LD, so this is the same. We are also producing antibodies like well-preserved peptides. That means peptides that we know that almost all living beings here on Earth have some proteins, and this protein has a small pieces, a small module of peptide that are well-preserved. And you will see later, we have produced against this peptide, and also we are able to detect them. Or we are also trying to understand what could have happened on Earth 3 billion years ago in the different type of life, by try to understand what proteins could be there by using ancestral sequence reconstruction and what we call protein resurrection. That means we can infer the sequence of protein that were 4 billion years ago, and then this protein produced in the laboratory and study the function of this protein. Okay, we can do that and produce the antibodies and try to find it. So the idea is that one here is to try to understand what is happening, traveling in time in the evolution tree of life, like you see on the right, infer what kind of protein could have been billion years ago, produce antibodies against this protein that we have somehow in silico produced, and in silico and in life, because we are producing in the lab, produce antibodies and later go to different environments and see if we can detect them. Okay, because the idea is life on Mars should have evolved very slowly since the beginning, because you know the temperature drops, no water, etc. So we don't expect a fast evolution. While on Earth we have a very fast evolution. So if we are able to find our proteins that we have inferred from this system that could have been in billion years ago, and we detected in extreme environment here on Earth, we can somehow use these tools for detecting these similar proteins to current Mars that shouldn't have been too much different from the older one. I don't know if you follow this. It's complicated, but yeah, but that's the idea, okay? And indeed, what has happened? We were very happy to find some of these peptides that we have inferred from a just in silico way by doing ancestral sequence reconstruction. And in that case, were with some protein that were done by colleagues in Granada. They give us the protein, we produce the antibodies, and these antibodies were used to test these samples that you have seen before in the extinguisher in El Tatio. And we detected one of the peptides, just a small sequence, 7 or 8 amino acids, but it was a peptide that was generated artificially from our ancestral sequence reconstruction. So that's, that's one of the key points. So a peptide that was artificially generated is there. So it's something that's the idea, okay? And that's why we are doing this work by Rita that is now accepted in Journal Molecular Biology. She has done ancestral sequence reconstruction of the growing, chaperonins. It's a kind of protein that is all over life, from the simple bacteria to humans. It's a very universal proteins that make this wonderful kind of rings that you see here, and helps to other proteins to fold properly. And this one here you are seeing is the structure that she has generated that theoretically has 4 billion years. Okay, so you can imagine it's not real, but it must have 4 billion years. In spite of having only 60% identity with the true proteins, they fold exactly, it form the same structure as the true ones. So that means the reconstruction is relatively well done. So, okay, we are doing some study with this, and the idea is to try also to use this, and we have already produced antibodies, and the idea is to use these antibodies to try to find in the extreme environment on Earth if we are able to distinguish some specific particularity only in ancestral reconstructed proteins, okay? And the other strategy I told you before is, okay, we use very well preserved peptides that are in many proteins here on Earth. Because the protein, you might have proteins that may have different functions. But as key part of this function, just imagine a house. Every house could be different. I could have many inhabitants, whatever, but the kitchen is similar, or the bathroom is similar in all of them, okay? So that's what happened with peptide. There are many proteins that share common peptide. You see many of you for sure are not familiar with this, but what is called the helix-turn helix is a combination of amino acids that make this basic form for life. It's a kind of turn, you see. The beta propellants, these are basic structure of peptide that are in many proteins. So what we have done is we have produced antibodies against these peptides, and we have been able to detect, as you can see at the top figure, we are able to detect even with this antibody common structure, 3D structure, even the peptide has different sequences. We can have different sequences of amino acids, but they fall the same way, and we detect the 3D structure. So it's one of the good things we can do, which is great because it make us more universality to our antibody. And in the B part, you see, is what I told you before. We are able to detect these peptides when they are bound to minerals. So we don't need to extract from the minerals because sometimes it's very complicated. It's practically impossible to extract many organic compounds from minerals. They are so bound and so sticky that it's impossible. So in that case, not always, minerals are our friends because they allow us to detect these peptides in a sandwich type of way. And yeah, but as I said you before, this brings us to the question, are amino acids and monosaccharide good biomarkers? It depends. It depends. The composition, the variety, etc., but not necessarily. As you know that meteorites has a lot of amino acids, and they are there, and perhaps one of them, I don't think it has been detected tryptophan. Perhaps it's one of the most rare amino acids in meteorites, but you know, there has been detected uracil and many other compounds. So it's very difficult to show that. Okay, so what we have done is to go a little further. Okay, amino acid perhaps not, but let's put a sequence of amino acid. Let's put 2, 3, 4, something like that. So we have done, what we have done is to synthesize these amino acids in the laboratory. Some of them has been already produced in a prebiotic experiment, but the idea is to use another immunoassay system, what is called the inhibition immunoassay, where what we do is just to immobilize a complex where we have this structure, these amino acids are similar, okay? And we incubated one of the sample with fluorescent antibodies and another one with the sample. If there is no these yellow dots in the samples, like in the case, the upper case, the antibodies bind to the complex. So we have 100% signal in our case. When there is something in the sample that compete and bind to the antibody doesn't bind to the complex. So we have a loss of signal. So this is a different assay that is called for loss of signal. It's a little more tricky, but it's wonderful because many commercial immunoassays uses this system. And we have done this with this peptide. We have produced antibodies against this peptide. You can see here this a peptide with 10 glycines, 10L aspartic, DS aspartic, 10D, the stereoisomer, D dextrohero and levohero, you understand? D aspartic to the right, L DG DG DG, so pentamer, D DG DG, pentamer, and the heptapentide of proline. Proline is practically impossible to find in a prebiotic mixture. But you can see how we can detect specifically on the right. You see the anti-LD peptide, anti-L aspartic, anti-D aspartic. You see how the places is in different ones. So that is a specific detection. And not only that, we are able to detect in the case of anti-L aspartic, we only can detect the 3 amino acids at the end of the peptide, not only in this peptide or whatever other construct. We have tested many of them. So the point is that with this peptide, we can detect even the best cases. We have D aspartic, 3 aspartic very well, and many others you can see. We have, I'm not going to detail, but this is the quantification of all of them using different target molecules. To explain one case, for example, here on the first one, you see these are inhibition curves. That means that in those places where we can detect any loss of signal, that means that it's not working. Okay, and we tested, for example, in the first case, when we use as inhibitor, the G3, that is the glycine with 3 amino acids, G4, 4 amino acids, G10, okay? Or when we use, let's see the most, the anti-L aspartic, you see we can detect 10 minus 2 minus 1 molar. So I'm using D6 or D10. So the idea is just to narrow the detection. Okay, I don't know how, 5, okay, so well, the idea is to have a collection of antibody to cover the most universal set of biomarkers as possible. Okay, one way to overcome the criticisms that I always have, that this is too terrestrial. So you can imagine we have also proposed SOLID for several missions, even proposed for Mars 2020. We proposed for the discovery calls, NASA discovery calls in the proposal led by Chris McKay and NASA Ames that is called Icebreaker. The idea of Icebreaker is go to the northern terrain where the Phoenix lander took place, make the drills. That's why the drills we are doing there. Make the drill, take the sample, and one of the instruments to analyze is the solid that you see there in the platform. We also applied to the Icebreaker in the 2019 call. It was very well evaluated, but by then they preferred to choose the mission to Venus, I think. So in the end, I think we are still on this. But okay, it was well. And one of the things what happened is that the Icebreaker mission was now adopted by JPL. And the mission has passed through to call Mars Life Explorer. And now it's in the Decadal Survey of NASA. Perhaps some of you have heard about it. But now, instead of being a 500 million mission, now is at least 2,000 million mission. And practically, and instead of going to the northern terrain, the plan is to go into the more equatorial places where there are some places where ice at 1 meter depth, something like that. And the idea is practically the same. It's using the same platform, and but so far we don't know if we can go there or not. But it's a mission, you know, there are a lot of uncertainties now on NASA, and who knows. But the idea is, if we have any chances to be there. And finally, it's just, okay, what's the future? Okay, the idea is just to be also involved, if we can, in the new ESA mission to Encelado, the ESA-L4. So this is just a couple of three slides from Florian Kelt, who is involved in the payload working group, where the idea is to make a lander, to take samples, drilling, scoping, or whatever, extract the sample. So I hope they can do for us. We don't need to use our sample preparation unit, and do some pre-characterization and then some organic analysis. Okay, and in the report they mention several instruments, etc. And one of the instruments they mention is this, like this slide here you see, where there are some immunosensing platform like the life marker chip that perhaps some of you can hear about it is, well, it's similar to, well, it's the same as SOLID, but it's a long story that I will tell you all the time. SOLID for sure, and Pleiades that I don't know exactly, well, it may work or not too. But this is the idea, okay? For sure, for Europa, our SOLID instrument now weighs 7 kilos. 7 kilos is a little bit complicated. And the concept is 800 and is encouraging is to use lab-on-a-chip concept for sure. Lab-on-a-chip concept is, you know, small chips with microfluidics, chamber cells, etc., etc. Yeah, and for sure it's an idea because miniaturize a lot, but clogging is always a nightmare. People that work in the microfluidics, clogging is okay. You can do microfluidics in your lab because you are handling and controlling a lot of things. But when you send something millions of kilometers apart, you have to have a lot of redundancy or a very robust way of avoiding whatever clogging in your channels, etc., etc. So that's why one of the concepts we are thinking is just to be even more simple. Avoid as much as possible pumps, valves, channels. Avoid as much as possible because every of the component has a possibility of failure. And what we propose is just, let's say, is the idea we are working on it is just using lateral flow immunization. Using, instead of putting the strip you see in the COVID test, put an antibody microarray and let the liquid go through. For sure, we will not control the velocity, etc. But we can control somehow by intelligent design of the strip where the liquids are going. So this way we avoid pumps, valves, etc. One of the weaknesses of this is the sensitivity is not the same. So it's something that we have to work with, and we are thinking of using electrochemical sensors. We are working with some colleagues that can help us on this, and yeah, the idea we are quite confident that we might have some. So it's very light, and you can have a lot of redundancy, etc., etc. But okay, just an idea. Let's see if and yeah, we have already started to work on this. But for sure, there are many challenges. We need to understand much better the environmental constraint from the immune. So planetologists here in the room might help us on this. Temperature, radiation, energy, whatever. We need to find a way to protect the antibody for years-long missions. We know that they can stand quite well for a 2 years mission to Mars, but for Europa is another story, or for Enceladus is another story. Sensitivity, we have to work, as I told you before, we have to see what's the best system we can have to keep similar performance as we have with fluorescence, because fluorescence is the best. Fluorescence is so far, for me, no level-free, no. Fluorescence is the most sensitive and specific so far. New materials, perhaps we need to develop new material for the systems, because paper, now for lateral flow, they use paper, different kind of paper, but is that multiplexing, we have to see the way how to put many of the largest number as possible of antibodies. Perhaps the idea we can make sandwich inhibitor immunoassay, both things. That's the good thing of having a light system, a very light system. And new antibody development, for sure. I think the idea is to be able, that's why we have initiated the line of producing antibodies against prebiotic compounds, because the idea is to deal with the result we might find there. Just imagine we go there is a complex prebiotic chemistry, but there is no life. Just there are some amino acids, some peptides, if you detect this amino acid, it doesn't mean there is life necessarily. If you are able to detect some D and L peptides, why these D peptides? Perhaps it's an strong indication that it's a prebiotic mixture only. But okay, so that's the idea. And I don't want to go any further because, well, there are many things, but I hope you have had a general idea of what the project consists of. And just to mention the people that is now involved in the current project, there have been many people in SOLID along the history since 2001. But just mention the ones that has been involved in the current funded project, and some of them are Pedro, Eduardo, and Paulo has PhD that has now writing. Maria Angeles has left. A new incorporation, Silvana, and the rest, perhaps you know all of them: Mercedes, Laura, Miriam, Sergio, Miguel, Rita, and from robotics, Javier, Julio, Ivan, here, Alberto, Raquel, people from NASA Ames, SETI, and other collaborators we have. Okay, and just thanks to funding sources, because they are the blames. If they don't pay, we can't do anything. Okay, so thank you. Thanks.
So thank you, Víctor, for sharing with us your research and with your team. And for the ones that already hasn't sent us their availability and want also to follow the lead of Víctor and want to share with us their research line, please do it.
In 45 minutes, please.
Now, if you have any question about this.
Thank you, Victor. Yes, thank you for the nice talk. I have a curiosity. So you mentioned that you are synthesizing these proteins, no, in the lab, so that you want to test whether having this generic budget, no, of proteins, whether Solid could detect them with the antibodies. Then I was wondering how this would work, having these proteins and their extreme degradation. So you mentioned, no, that these biomolecules experience this strong degradation. So how would Solid react after these proteins have been subject, no, to this to radiation and so on?
Yeah, no, it's a good question, because this is one true serious problem we face it when we take environmental samples from we have taken some from 200 million years, that those of Atacama, 200 million years, and we have done extraction and analyze the protein there. We identify the presence of peptides, okay? But the entire protein is very difficult. The entire protein, perhaps, once, unless you have a very well protected settings where the protein has been kept wonderful. Indeed, there are some colleagues at North Carolina. It was a woman here some time ago that she has found I don't say intact, but almost intact proteins and DNA in dinosaurs fossils. So it's amazing. No, but it's not the best. So you are right. What we expected in nature when after millions of years is just the presence of peptides. And these peptides usually are bound to minerals. That's why the importance of using even minerals to detect them. But the good thing is that the antibodies don't detect the entire proteins. Antibodies never do that. Antibodies are another protein, but bind only from 3, 5 to 10, 12, 15 amino acid places only. So that's our strategy, just to try to identify what kind of peptides are well preserved in nature, and then phase to detect them. But yeah, for sure, and one way is that one is just, okay, let's imagine on Mars 4 billion years ago, if there was some light, has been similar to that on Earth. If protein on Earth looks like this, we might have. So it's for sure, it's as we say in biology, it's a shotgun strategy. It's like sequencing. Sequencing is what is called a shotgun. You start to sequence a lot of things, and at the end you matches the sequences. So it's, yeah, but you're right. It's a critical point.
It was very interesting to see that 2025 P-STAR proposal. Can you elaborate, I mean, to the extent that you can on that proposal? Is it framed in, I'm guessing it's to learn lessons for a potential Mars Life Explorer-like mission? Is SOLID part of a like more complex set of instruments that are right now working on the Antarctica?
Excuse me, the question is for what?
The P-STAR proposal.
P-STAR.
The context of that proposal is to get lessons for a Mars Life Explorer-like mission.
The P-STAR proposal I show you here, yes, is focused to Mars. The idea is just to increase the maturity of the drilling system that is produced by Honeybee, and the connected to different instrumentation. In our case, they always count with us because we a good example of just taking the cuttings, put into SOLID and analyze, but also there are other instruments. This year was also a Raman instrument, and other times the capillary electrophoresis. And the idea is to put all together, okay? Improve the drilling system and fit other instruments to see how this project can help in designing missions to Mars.
And out of curiosity, is there a weather station on
We never, because they are very interested in the drilling, and we are interested only in SOLID. I don't think anybody has paid attention. I don't know, Michi, there was any environmental station on the rover? No, I don't think. I think Brian never paid attention to this. So far, he is also focused on the drill and yeah.
Any more questions? Yeah.
I have a political question.
No, question.
Knowing that Chinese, they are doing quite well in Mars exploration.
Yeah.
I mean, did you ever consider, or it's our relationship with NASA, which I really don't know the state at this time, to try to collaborate with them?
Well, it's a good point. Yeah, I never thought. Well, I thought about that, but the point is, we have been collaborating with NASA since, as you know, since 2003, making this change. Perhaps NASA can do whatever they want, but I don't know if the collaborators can do whatever they want, because they do whatever they want because they control what they want, what they do. But in our case, you know, but so far I just prefer to wait and keep collaborating and see what's happening. I don't know, in the MIPAG forum, things seems to move forward and to push for robotic exploration. I don't know. On the ESA side, there are some people that is now thinking in a post, let's hope, a post ExoMars mission, Rose Franklin mission. So, well, let's keep. But yeah, I don't know. Politically, is delicate.
Anyone else in the room have questions? So I think
No one else has a question? Okay, very quickly, what's your like honest opinion about the recent discovery by the Mars 2020 mission about the potential biosignature? I'm sure you read the paper, and what's your overall opinion about it?
Well, it seems to be mostly a biomineral signature. There are people that say that may be also produced by, as always, by a non-biological process. So it's what I read is that they have a strong correlation with the organic matter, global organic matter that has been detected by SERLOC, the Raman spectrometer. Honestly, it's interesting, but I don't know. This could be, you know, how is the word definitive. It's perhaps one of the things. I think that unless we detect something that wow, this is wonderful, some proteins and whatever. At the end, the presence of life on Mars or not would be you know, some addition of many proof, many things. I think this one is one. It's a good samples to bring back. That I guess one of the ideas of the paper is to this is an interesting one to bring it to Earth. Yeah, sure. But for sure, if the point is, I don't know if there is some mineralogist here, perhaps, or some can tell us these minerals they have found has anything that is only produced through a biological process. I mean, there is the production of sulfuric acid and SH2 and others. The only way that has to be produced there it was through biological process, or not? Perhaps not. That's the point. Or if the mineral phases are specific for biology. I don't know. Perhaps they have not been able to do that. Because you see, one example was the ALH 8000, the meteorite from 1996 from the Mackay. The magnetite, this magnetite, they needed a lot of analysis to conclude that that could have been produced from a abiotic process too. So complicated. If we detect some proteins, perhaps yes. Even.
Yes, please.
Okay, my question is, you have been very focused on Mars during these years. So you work with very specific terrestrial analogs and molecules that you consider that are stable in the conditions of in terrestrial conditions and maybe on Martian conditions. But now you are moving to the icy worlds. And my question is, have you started to work with different terrestrial analogs or to consider laboratory experiments to constrain the stability of the molecules, considering that you have other factors that can affect your molecules, like, well, if you have the molecules in the E-ring of Saturn or something like this?
Yes, indeed. Part of the PhD thesis of Sergio is on this direction. It's just to move through Europa or Enceladus analog composition and do at least some laboratories in some experiments in the laboratory to understand this type of thing. What kind of biomarker we can find, even try to see if under this condition we can have some prebiotic production, just feeding with some amino acids and see if we can, by doing different experiments of wet dry that we have already started, we can find something new and understand the stability. Yeah, sure. It's something that for sure we can. Indeed, we are waiting for somebody that gives us the right composition of these salts and see what happens. Yeah.
With the minerals and in Enceladus, you will have problems with
salts.
With salts and with ice grains, because the organics are very stick to the ice grains.
Yeah, but I guess if they are melted, the organics will release or not.
Salts. Yeah. Yes. I mean, you have different problems that you have to solve now, and you can combine your instrument with
others,
potential others that could fly.
This is for the next project that I have to apply. Yeah, no, it's true.
Okay, I think we have a question online.
Hello. Yeah, we have here some questions in the Zoom. Jose Caballero asked 2 questions, but he said he has to go, so he will ask you in person. And on the other hand, we have Ignacio Morales Gil. So is there any risk of contaminating space bodies with the antibodies in remote operation?
To contaminate, for example, Mars. Yeah, well, this is something that in all the proposals we have applied before, this is something that we have to deal with. So we are in the, I don't remember, in the class 4 mission, I think, is one of the most restricted for this type of things. Everything has to be encapsulated in some compartment that has not any chance to go outside, etc. So yeah, this is something that is always in the protocols for doing this mission, and the idea is always to encapsulate not only our antibodies, for sure that in our cases they are very confined in a very close compartment. But for example, the others for the drill or the robotic arm, all this type of thing has also to be considered for planetary protections. Yeah, in the case for at least for forward contamination, it's a problem because there might be other instruments that we have to avoid that, mainly because we don't want to contaminate to other instruments that they detect something that is come from us. So this is important, has to be very close.
Yeah, of course. Thank you. And then the other question is from Jacobo Aguirre. He says, thank you for the talk. And the question, in your opinion, in summary, which would be, you think it is the most difficult challenge that you face regarding sending solid in a spatial mission, and how to overcome these problems?
The most difficult one, the most difficult one is perhaps having or finding a mission that really wanted to detect, to search for life. Is the most difficult, because the point is honestly, I was a little bit disappointed when I saw the objective of the Mars 2020 objective, because in the end they didn't want to search for life. Now is a good one. For example, the Enceladus, the L4 mission is wonderful because they really wanted to search for life. And a part of that, for sure, is there's a technological, I would say, a challenge for sure, but it's something that can be solved if you have a mission, just dedicate time and effort to do that. But basically it's to fit in the objective of a mission. But because many of the critics we have had, for example, in the icebreaker, there was no significant critics for SOLID, for example, and a part of what I told you before, that we are too Earth-centric. But not much critics on that. But the point is there a mission for searching for life? And later, for sure, we don't know the competence if there is one where we can go with others. Yeah.
All right, thank you.
Okay, if there are no further questions, thank you very much, Victor.
You're welcome.
And thank you, everyone. See you each Thursday at 12. You know, we will have mostly in-house seminars, but also we will have few from outside. So if you have any suggestion or you want to volunteer for give a talk, please contact us.